Hi everyone, my name is Mao and I work for Dr. Jean-Luc Janik at Cornell. Today I will share with you some unique aspects of breeding for sugar kelp, Saccharina latissima. So, kelp has mainly been used as a food source for human consumption, mostly in Asian countries. Lately, in the US and in the European countries, more research are interested in breeding kelp for biofuel. People also extract compounds from kelp for pharmaceutical uses. The selective breeding on Saccharina japonica, which is one type of kelp, started early in the 1940s in Asian countries. However, kelp breeding in the US is still at its infancy stage. In 2018, our collaborators went out to collect wild samples from Gulf of Maine to southern New England. These two regions are separated by Cape Cod Canal, which is known as a biogeographical barrier for many other species. We did find very distinct subpopulation structure between these two regions, and the Gulf of Maine population is the focus of today's talk. Kelp has a unique biphasic life cycle. We start from the sporophytes harvested from ocean. These are diploids and can be phenotyped. Once sporophytes matures, they produce source tissue, which is the reproductive tissue. We collect them, let them induce spores in the water. These spores are free-living and floating around in the water, which is very different than row crop seeds. We have these spores go through flow cytometry single-cell sorting system. Each spore then can develop into either a female or male gametophytes. Gametophytes are haploids. We store each gametophyte in a separate container, and this is our germplasm stock. When crossing, we mix them, and if fertilization happens, young sporophytes will develop, and then we spray them onto seed string. 4 to 5 weeks later, we transfer them onto larger growth rope and plant them on the farm. This is what it looks like with our current breeding pipeline. We start from the wild sporophytes, which is our founder population. From there, we generated this stock of gametophytes, which is our germplasm. From that germplasm, we choose gametophytes to make crosses and testing the progeny sporophytes on farm. If by the time of harvesting these sporophytes are mature and reproductive, we can then collect source tissue and induce spores and to get the next generation of gametophytes in the lab. This gametophytes takes at least a year to grow and accumulate biomass in order for us to make crosses for the next generation. So our current breeding pipeline takes at least 2 years to finish a breeding cycle. Genomic selection happens for this gametophyte stage where we genotype them. We use the field data from sporophytes to build a model and predict the breeding values for gametophytes so we can pick the best gametophytes as parents to make crosses. Some pictures on how the pipeline looks like. Here is when we collected source tissue, clean them, and then let them induce spores in the lab in the water. Once they're sorted, the gametophytes can happen, and this shows gametophytes are stored in flasks with seawater. And then when we cross, we mix those tiny amount of female and male gametophytes, mix them, and then store back into flasks. This is after the mixture was already sprayed onto the seed string. Then the seed string were transferred to growing rope and ready to be planted in the ocean. Finally, the crew is harvesting. We used a very typical augmented design on the farm, including line effects, block effects, and year effects, using common checks across different blocks. Once we harvest this, we would measure plot level biomass traits as well as morphology traits, which indicates the blade length, width, thickness, stipe length, and stipe diameter. This is what it looks like. When we harvested the kelp, we store them in this huge container. And then we weigh each plot to get their plot level wet weight. We then cut it to get subsamples. We also look for source tissue and store them. For those individual blade measurements, we would keep the samples in the bags first. And After we finish all the plot level measurements, we then go to each one of those bags and then start doing individual measurements. So using these traits from sporophytes, we built genomic selection model and we evaluated the accuracy between different populations. In this case, training populations are data from year 2019, 2020, and year 2021. It's used as the confirmation population. We also try to predict between 2019 and 2020. So we use the pedigree and also marker information to build a relationship matrix. The total individuals that's included in this analysis are 950. So this would include founders, gametophytes, sporophytes, and the new generation of gametophytes. We compared different models to test the genomic selection accuracy. The first one is the base model, GBLOB model. We used both pedigree and markers information. The second one is general combining ability and specific combining ability model that's constructed in BGLR. We compared using pedigree plus markers to construct the relationship matrix versus only using the pedigree. In our case, we have both pedigree and markers estimated relationship matrix. We combine them using the Actemir et al. Method. In this figure, for example, he has 2 pedigree matrix and 1 marker estimated relationship matrix. Your output is a combined kernel. So in our study, we have pedigree estimated relationship matrix for all the individuals, and then markers estimated relationship matrix for the founders and for the gametophytes. We then combine all of them using the package. Here is trait heritability figure. X-axis represents different traits. Their abbreviations are listed on the right side. Y-axis is the trait heritability. On the left side of the dash bar here represents biomass traits. On the right side represents morphology traits. We do see that morphology traits have higher heritability than biomass traits. The first trait, dry weight per meter, which is the yield that we're interested in kelp. We do see that heritability for yield traits varies from one year to another, which is also typical in row crops. And we had tested significant year effects and genotype by environment interaction. We then looked at the correlation among these traits. Again, above this dash bar represents biomass traits, below it represents morphology traits. The first trait is dry weight per meter. Dry weight per meter is significantly correlated with the fourth trait, which is blade density, but not with any of the morphology traits, which indicates if a blade is very small, within that plot, those small blades may grow denser, and that in return contribute to the weight. For genomic selection, we first compare different models. On this figure, x-axis represents different models as I've mentioned earlier. Y-axis is the GS accuracy through cross-validation. The third one is general combining ability and specific combining ability model using both pedigree and markers information. It gives higher GS accuracy than the other two, though the difference is not significant. We then looked at the GS prediction accuracy between environments. Here, y-axis is the GS accuracy, and x-axis represents different traits. Again, we have biomass traits and morphology traits. Except for blade thickness, morphology traits in general gave higher GS accuracy than biomass traits between environments. The first one is the dry weight per meter. That's what we are most interested in. It had an accuracy about 0.17, which is on the lower range, but it's still within the range of GS accuracy for yield in row crops. We then added 2021 data as confirmation set. On this figure, x-axis represents different traits. Y-axis is the GS accuracy. Different colors represents training data set is from a different year or years combination. Again, the GS accuracy varies. And for the first trait, dry weight per meter, is around 0.2. It's still on the lower side, but that still falls within the range of GS accuracy, predicting between environments for yield in row crops. Many factors affect GS accuracy, and we assessed the trait genetic correlation between years. We do see variation for the same trait from one year to another. Between year 2020 and 2021, which is in gray bar, more traits had larger genetic correlation than the other years' combinations. This is likely due to that year 2020 and year 2021 had similar length of growing season, which is longer than year 2019. For dry weight per meter, we estimated almost zero phenotypic correlation between years, and so GS approach is still a little more predictive. We do think that the large year-to-year environmental variation and G by E is limiting our ability to selection. Let's take a look at the breeding values for dry weight per meter over generations. X-axis indicates 950 individuals. Each dot represents each individual. Y-axis is the breeding value for dry weight per meter. Different generations are shown by different colors. Black is the founder sporophytes. Green are the gametophytes from founders. These are also the parents of farm-grown sporophytes, which are in dark red dots. Then we had a small amount of gametophytes that are collected from farm-grown sporophytes as shown in red. Because gametophytes from those farm-tested sporophytes are collected at random plots, due to that source tissue ripened randomly on farm plots, we do not see any improvement of breeding values when you're comparing the red ones to the green ones. Although we did see some farm-tested sporophytes revealed some improvements as they show higher breeding values than the founder sporophytes. As you may have seen, there are many challenges that are unique to kelp breeding compared to the row crops. In the past 3 seasons, less than half of the crosses we made generated useful data. The plot either did not grow or if they grow, but they become very patchy. For example, you can see on the left side of the plots that they grow somewhat more uniformly than the plots on the right side here. We discard any data that come from a plot like those on the right side. Second, there is a huge randomness on whether or not source tissue can ripen when we harvest. This would affect whether or not we could select best sporophytes to induce spores and produce its next generation gametophytes. Lastly, the gametophytes that we maintain in the lab, they grow really slow. In order to accumulate enough biomass for making crosses, it takes a lot of efforts and time. It's very labor intensive to maintain those gametophytes in the lab. We need to change seawater every week or 2, and sometimes contamination could also happen in the culture, and then we lose that gametophytes. Here are some thoughts for next steps. We did a simulation study testing out different scenarios in affecting our gain of selection before. We find that high genetic gain could be achieved by shortening our current breeding cycle from 2 years to 1 year. We then tried a proof of concept experiment this past season and concluded that it is possible to complete kelp life cycle within a year on farm. Though we could only plant and harvest a few of these individual sporophytes, mostly still due to their parental gametophytes biomass limitations. Those sporophytes could not be tested as meter-long plots for yield, but we could incorporate them into our breeding pipeline as common practices for early-stage nursery screening step. There are other breeding pipelines that are worth assessing. For example, in order to overcome the gametophytes biomass limitation, we could make mixture plots where multiple female and male gametophytes are used, much like in forage breeding. Our goal is to select best gametophytes and recommend to farmers in making hybrid crosses. So far, we do not have much data on inbred lines, though inbreeding depression seemed very unlikely, so we need to continue our efforts in developing inbred lines, which could help reduce labor from a long term, as by then, we do not need the single-cell sorting step for the gametophytes. Thank you for listening. I'd like to thank my advisors at Cornell University, our collaborators at University of Connecticut, Greenwave Woods Hole Oceanographic Institution, and our funding source, ARPA-E. Welcome for questions or comments and suggestions.